We have developed an easy method to test food samples for the different C. perfringens toxinotypes. We are particularly interested in the prevalence of toxinotypes B and D, which produce the epsilon toxin. Our protocol allows easy enrichment and detection of the various C. perfringens toxinotypes without the use of anaerobic chambers or incubators with limited subculturing. This method could also be used to test soil, water, fecal samples, and more. Demonstrating the procedure will be Zuha Anwar and Samantha Regan, technicians from the lab.
Begin by preparing modified rapid perfringens medium, or RPM. Combine ingredients according to manuscript directions and autoclave the mixture at 121 degrees Celsius for 15 minutes. Allow the medium to cool to approximately 40 degrees Celsius. Then add D-cycloserine to a final concentration of 440 milligrams per liter. Aseptically transfer the RPM to 15 milliliter conical tubes and store at 4 degrees Celsius for up to 1 month. Prior to using, warm the medium to 37 degrees Celsius. When collecting samples to be tested, transfer the food to the laboratory according to manuscript directions. If not testing immediately, the food can be stored at -20 degrees Celsius. Make note of the type of food and the country of origin, as well as any other relevant information. To test the food, transfer approximately 1 to 2 grams to a sterile petri dish, and finally mince it with a sterile razor blade or scalpel. Inoculate it into 10 milliliters of PBS in a